Hello everyone, I'm Brittany, an educator from the DNA Learning Center, and today I'm going to be discussing industrial enzymology. Now the picture behind me on the screen is the DNA Learning Center, and I hope one day you'll be able to come and do a lab with us here on site. But for now, we'll do this virtually. What is industrial enzymology? Industrial enzymology is just the use of enzymes in industry, such as the production of cheese or the making of juice. But before we talk about that, let's just do a little review on DNA. So this is an example of the structure of DNA, which is a double helix. And DNA is important because it's the cookbook that makes us who we are. Since it's a cookbook, that means it needs to have recipes. But if I have a recipe for a cake and I want a cake, can I just say I want the cake and the cake appear? No, there's a process. There's other ingredients that are necessary. So with my double helix, I have a small segment of DNA, which is known as a gene. And this gene will produce a protein. And the protein, when it is expressed, will give us different traits. Now, what are traits? Well, a trait is just an individual characteristic. It can be something such as eye color, hair texture. There are many different traits, but all of it is due to our genes. Now, I want to spend a little more time talking about proteins. Proteins are very important and they serve a number of different functions. So there are structural proteins, and an example of structural proteins are actin and myosin. These two proteins are important for muscle contraction. For transport, one example of a transport protein is hemoglobin. And what does hemoglobin transport? If you said oxygen, you're correct. There are messenger proteins, such as the hormones, which allow different areas of the body to communicate with each other. And the focus of our discussion on today is that of enzymes. Now, enzymes have a number of particular characteristics. An enzyme can catalyze a chemical reaction. Now, what does it mean to catalyze a reaction? To catalyze a reaction means to speed up the rate of the reaction. This is done by lowering the activation energy. Enzymes are affected by changes in temperature and pH. This is very important because these two items can denature or change the shape of the enzyme, which creates a problem since enzymes fit only specific substrates. So if the enzyme denatures and no longer fits its substrate, are you going to get the expected reaction? No. Enzymes, as you know, are proteins, and they usually end in Ace. If you watched any of our other labs, you may have heard us talk about lactase or catalase. You may have heard of polymerase, but we're going to talk about different enzymes on today. Now, I don't want you to get confused. There are exceptions where enzymes don't end in Ace, but for the most part, if you see an Ace ending, you know that it's an enzyme. And then during reaction, the enzymes remain unchanged. This just means that you can use that enzyme in another reaction. What this screen is showing you is what we just discussed. So you have an enzyme and it joins with a substrate. And what happens is in a digestion reaction, the substrate, which is just the molecule the enzyme acts on, is broken down into two smaller products, product 1 and product 2. However, enzymes can also have a synthesis reaction where two products are acted on by the enzyme to create a new product or substrate. Now, do you remember when we talked about the two factors that can denature an enzyme? What are those two factors? That's right, pH and temperature. And this just shows if an enzyme is denatured and the shape is now changed, it may not, or in this case, it's definitely not going to fit its substrate, and you're not going to get the desired product or result. Now, enzymes are very important. We know they're involved in digestion, but it's important to differentiate between the two different types of digestion that does occur in the body. So there is mechanical digestion and there is chemical digestion. Mechanical digestion is just the physical breakdown of whatever product it is into smaller versions of that same product. With chemical digestion, what happens is the compound actually changes. So it's broken down from one molecule into another molecule. And examples of this are amylase. And amylase is the enzyme that breaks down and acts on starch. There's lipase, which breaks down lipids. And there's proteases, which, as their name suggests, breaks down proteins. So, it's a very interesting picture that's on the screen behind me, but very shortly you'll understand why. So we're going to talk about one of the ways enzymes are used in industry. And to do that, we need to go back in time. So thousands of years ago, nomads had to travel across different locations. And in this scenario, we're talking about traveling across a desert. Now, if you're traveling across a desert, what would you need to bring with you? You would need to bring water. Did they have the fancy water bottles that we have now? No. Could they carry it in a basket? No. What they did is they actually used the organ of another animal. The organ is the picture you see behind me. What organ do you think they used? A stomach. That is correct. So what they did is they would clean out the stomach because they weren't going to drink the gastric juices, and then they would put the water into the stomach and carry it across the desert. And then when it was time for them to drink, they had water. They decided to take this a step further. If I can carry water this way, why not carry milk? So what they did is they poured the milk into the container and they tied it off at either end and they made their trek across the desert. However, when they reached their destination and it was time for them to take a drink, they poured out what they thought was the milk and realized it was no longer the white color. It was this almost clear yellowish color. And the stomach still had weight. So when they cut open the stomach, they looked inside and there were these little white clumps. Do you know what they found? They found cheese, or more specifically, curds. You know the nursery rhyme, Little Miss Muffet sat on a tuffet, eating her curds and whey? They found curds and the liquid whey. Now, how? Well, what happens is we know that milk has a creamy texture. The reason milk has this creamy texture is what happens is the casein is separated by these particles which separate them within the milk. However, what happens is an enzyme known as rennet, which is found in the stomach, actually breaks down those particles and the milk clumps together. So even though they were removing the enzyme, they couldn't get all of it out. So the enzyme was there, they had the milk, but there's one other factor they had. What other factor is that? They had the heat. They were traveling across the desert. So the heat acted on the enzyme, which then acted on the casein particles, kappa casein specifically, but that's not important for this lecture, and resulted in the curds and the whey. So what we're going to be doing in this experiment is we're going to be doing the same thing. And we're going to be using emparase. As you see right here, it says rennet, but emparase, it's called emparase just because an emperor actually utilized this science to help produce cheese. Now what I have over here is milk, and it's at about 55 degrees, and I heated it to that temperature because I needed to simulate the appropriate temperature. I have my emparase in these little microtubes right here. So I have my enzyme. But what factor am I missing that they had when they were traveling across the desert? I'm not using a stomach, so I'm missing the pH. Now this milk is a combination of buttermilk and whole milk. And the buttermilk is acidic, so it's going to help create the appropriate pH environment that I need for this experiment. So what I'm going to do now is I'm just going to take off the tinfoil and I'm going to pour equal amounts into each cup. Now I have a positive cup and I have a minus cup. The positive cup is for my experiment, where I'm going to be adding the enzyme. The negative is for my control, so you can see what happens when I do not use enzyme. All right. So what do you think I need to do next? I need to add the enzyme. So I'm going to take my Emparase, and I'm going to use a dropper. I squeeze the dropper, I insert it into the tube, and I take up all of the Emparase. I no longer need this tube, and I'm going to drop it in my waste beaker. Where am I going to add this Emparase? The plus or the minus? I'm going to add it to the plus, or my experiment. And what I'm going to do at this point is I'm going to let that sit for a while, so that way the Pectin Ace, that way the Emparase can work on the milk, and then we'll take a look at that in a little bit. But what I'm going to do now is switch to another enzyme that is used in industry. The enzyme that we're going to be talking about now is that of Pectin Ace, and it's used in the production of juice. My first question for you though, what is the purpose of an apple? Some students would say the purpose of an apple is for us to eat, but is that the tree's purpose for creating the apple? No, it's not. The purpose of the tree producing the apple is to produce more trees. Now, what happens is when the apple is eaten, the apple has a very hard outer surface. If you've ever cut open an apple and looked at it, you can see how tough the outer surface of the apple seed is. However, when it's consumed by an animal and it passes through the digestive tract, it's the perfect environment, because then when it is now back into the environment, it's already in fertilizer. But not all apples are eaten. In fact, I'm sure you've seen some apples that have fallen from a tree and are a bit mushy. Do you think that the animals are going to be eating from that apple? Probably not. However, what happens in this scenario is that the apple is acted on by a specific fungus. This is Aspergillus niger. This fungus actually releases Pectin Ace, which breaks down the outer pectin to release the juice of the apple. That's why some apples from the same tree that have fallen, some stay firm longer and some stay firm for a shorter amount of time, because the apple is designed to be able to make it maintain water longer or a lesser amount of time. So in the case of apples, what you see is the juice is held in by the protein pectin. And what happens is when Pectin Ace, it has the ace ending, so we know this is the enzyme, acts on the pectin substrate, the juice from the apple is then able to be released. And what happened is it was realized that this could be utilized in industry. But before we go ahead and do this experiment, let's check the results of our last experiment. So what I'm going to do is I have my positive, or my experimental cup, and I have my negative, or my control cup. What I also have are 2 cheesecloths that I'm going to put over an empty cup that I have for each. And I've labeled them to help with knowing which is which. And so now I'm going to look at the change that has occurred between my experiment and my control with the use of Emparase. Now to do this, what I'm going to do, you want to make sure you do the same thing with both cups. I'm just going to stir it. To help it along. And if you're looking, can you see the difference that's occurring in the 2 cups? What is happening with my experiment? The color is changing, and when you even look at my 2 sticks, you can see that one already has cheese. But let's just go ahead so we can fully see what's transpired. So for my minus cup, I'm going to go ahead and use the cheesecloth, and I'm going to pour the milk into that cup. Now, it's possible to still have a little bit of cheese because the temperature could cause the milk to form cheese. But which will have the higher yield? That of the enzyme or just the milk alone? So I'm going to lift my cheesecloth. I actually need to squeeze it a little bit. And what you'll notice is even in my control, there still is a little bit of cheese. But let's check out the experiment. Now I can see now, I already can see the whey. Can you see? So I'm going to pour out the whey. It looks like I didn't even really need the cheesecloth in this case, but you see a huge chunk of cheese. So with the help of Emperase, we were able to speed up the process, which makes sense since enzymes speed up the rate of the reaction. To give us our curd and our whey. And you can see the color difference between our experiment and our control. So which in this case gave us a greater yield? I would say that of the enzyme. So now let's check out the same or a similar experiment, but with the use of Pectin Ace, because Pectin Ace acts on pectin. The way we're going to do that is by utilizing applesauce. I have already labeled my dishes. Now, normally when I do this with a class, there's a lot of students who do this with me, so the result is more dramatic. So in this case, I'm going to do it twice just to help you along. So I'm going to take and do about 3 spoonfuls of each, just to be equal. And as you can see, this is regular applesauce. And what we're going to do is we're going to test which gives a greater yield. Is it the mechanical digestion, or is it the chemical digestion with the use of enzymes? There we go. On this side of my rack, I have Pectin Ace. And Pectin Ace is the enzyme that breaks up the pectin from apples. So what I'm going to do is I'm going to use a dropper. And I'm going to drop the Pectin Ace into my plus whey boats, which are for chemical digestion. So I'm just going to take one, and I'm going to put it here. I'm just going to do this quickly to help with the process. And I'm going to take my other tube, which is also Pectin Ace, and put it into the other experimental whey boat. And as you can see in all of the trays, there is some juice already starting to form. So it's not abnormal for that process to begin on its own. However, Our question is which will give the greater yield. So now I'm going to let those sit, but I need to do mechanical digestion. For that, I'm going to take two popsicle sticks, and I'm just going to mechanically digest them for about a minute. And keep in mind, even though I moved the dropper through the positive whey boats, I only did it briefly. As you can see, I'm doing this side much more rigorous. And even while I'm looking at this, if you look at the experimental whey boats, what you can see is you can already see more juice spilling out around it, even though I'm not applying pressure to it. Okay. And now let's see, which do you think is more effective? Do you think it was the mechanical digestion or the chemical digestion? So what I also did to help us out is I pre-labeled cups. So I will pour my control in here, and I will pour my experiment in here. Now these are the biggest coffee filters I have ever seen, but they are perfect for the job. And actually, let me just go ahead and use these cups instead. So I'm going to go ahead and do the mechanical digestion first. If you do end up doing this lab at home, it does get messy. Make sure you have napkins and paper towels to help you out. I want to make sure I get all of it. And now for the other mechanical digestion or control plate. And now I need to get it quickly before it decides to run. And what I'm going to do is now I'm going to squeeze to see the juice. And when you're doing this step, you want to be careful because you don't want the coffee filter to burst because it can be fragile. So you want to do this step slowly, but pay attention to when you feel as if the coffee filter is going to break. And if it does break, it's okay. I've done that before. Things happen, but just do your best. I'm just going to squeeze a little more. I think there's still a little more juice in here. Okay. Whoop. And that is exactly what I just said could happen. All right. So now let's check out our chemically digested applesauce. You're going to do the same thing as before. Coffee filter. And you can already see the juice Weigh boat number 1, weigh boat number 2, and now we juice. And pay attention to which cup has more juice. Now, when you do this with a whole class, the results are more dramatic, but I think you can tell which type of digestion gave me a greater yield. Was it the experiment or the control? Can you tell which one is higher? The experiment gave greater yield, which is why this is also used in industry. Now, one thing I want to mention is that to help you with understanding that an enzyme can be reused, this morning I actually did the same experiment. And what I did is what was in my experiment and control tubes, I poured it into 2 different tubes. Now, do you see the color difference? When they started, they were the same color. However, the plus tube, which had the enzyme, is now more clear because the enzyme continued to work, whereas the mechanical digestion stopped where it was. So if you're using a large amount or want to make a large amount of juice, which would be more effective? Having the enzyme which continually works or mechanically digesting? I think it's clear to say it would be the use of enzyme. So I hope you have enjoyed this journey through industrial enzymology on today, where we were able to turn milk into cheese. We learned about curds and whey, and we learned a little bit about the process of making juice. Thank you so much for joining me on today. Feel free to check out any of our other videos on YouTube for DNALC or check our website as well. Have a great day.